Hello, so for anyone who doesn't know me, I'm Danielle and I'm a third year MPhil student. The name of the project that I'm going to be working on is Forensic Colour, where the main aim of our research is the identification of pigments, and in particular the ones that have been used in artwork. So before I go into why this is important, let's start with what the project is all about. Colour. When it comes to producing colours that we can see, there are two different methods, additive and subtractive. The additive mode is when certain wavelengths of light are mixed to create colour. As you can see in the diagram below, the three primary colours of light mix to make three secondary colours. Additive colours will begin as black and will become white as more of the three primary colours are added, meaning that with these three primary colours mixed in different ratios, you can make all of the colours that we can see. When it comes to subtractive colour, this begins as white light, and as light is subtracted, so filtered out or absorbed, this creates the appearance of colour. Because we're going to be looking at pigments used to produce paintings and artefacts, we're interested in subtractive colour, which makes sense because pigments allow us to see colour when they are illuminated with white light, because they will absorb some portions and reflect others. So, how are we able to see colour? Well, the human eye contains millions of photoreceptors called rods and cones. As we can see from this cross-section of the retina, there are three different types of cones in the eye detecting red, green and blue light. So going back to what we just looked at, the human eye uses additive colour to see. Depending on the intensity of light detected by each type, it allows us to perceive and differentiate between 10 million different colours. Colour blindness happens when two peaks of the absorption spectra of the different cones aren't distinguishable. So this is very common with the green and the red cones. Rods are reserved for when light is very dim, and because they only detect intensity, this leads to us seeing in grayscale when it's dark. Although the way the human eye works bears many parallels to the workings of a CCD, with the cones acting as different colour filters, the perception of colour is much more complicated. It's not just what the eyes detect, but what the brain makes of this information. You probably remember the photograph of the dress that went viral a few years ago, where some people saw it as blue and black and the rest saw it as white and gold. This is due to the phenomena called colour constancy. The perception of colour is not just a function of wavelength, but is dramatically influenced by your past visual experiences. With the dress, it all comes down to what sort of conditions your brain is accounting for. Unlike a camera, the brain won't take the image at face value, leading to such a dramatic difference in perception in this case. We're not very good at determining absolute hues because our brain takes into account the colour of the light illuminating whatever we're looking at. It's the same with the dress. Depending on what colour you assume the dress to be illuminated by, you'll perceive it differently. So out of context of the dress being under lighting, you can see that the stripes are clearly blue and black. Okay, so bringing it back to the motivations for our research, the main application of pigment identification is in art history, where being able to identify pigments not only aids people in knowing how to preserve and restore paintings and artefacts, but can contribute to being able to date and authenticate pieces. Further to this, knowing what pigments were used to produce a painting, for example, could help to identify who may have painted it, where it could have been made, and like we hinted before, when it was made. There have been cases where being able to identify the pigments has meant that we know things have to be copies, because while some pigments have been around for thousands of years, others have only become available in the past few centuries. Titanium white is a really good example of this, because it was only discovered in 1821 and hasn't really been used in paints until 1921. Okay, so now let's look at a specific case where using pigment identification techniques to analyse a painting has been crucial in its restoration. This is a 1642 piece called The Night Watch. It was painted by a Dutch painter, Rembrandt, and is one of the most famous paintings in the world. Operation Night Watch is a restoration project that took place in 2019 in a museum in Amsterdam. The backbone of the entire project is the use of the most up-to-date scientific techniques to determine the composition of the painting in fantastic detail, and hence to restore it in the best possible way. Because this was such a huge project and involved the collaboration of scientists from around the globe, the techniques used are a brilliant representation of all the analytical methods available to us today in the art conservation world. There are a wide range of imaging techniques used, as we can see here, including UV fluorescence and reflectance spectroscopy, that are performed in a portable manner, so without moving the painting itself and without any destruction of the painting, And down here, we see some tools that we used on samples that were taken from the painting, including light microscopy, Raman spectroscopy, and mass spectroscopy. When it comes to the different methods of analysing artefacts, the Night Watch really highlights some of the qualities that we want. Firstly, we want these methods to be portable. Rembrandt's painting is not only old, but it's huge. It's around 3.5 by 4.5 metres and weighs 337 kilograms, so moving it not only risks damaging it, but is extremely impractical. Also, mass spectroscopy, along with others mentioned above, require a sample to be taken from the painting. They're very effective in determining what the painting is made up of, but differing from optical techniques, they're much more invasive and destructive. So I'm now going to hone in on the reflectance spectroscopy and Raman spectroscopy methods. So I focus on these is because firstly, they're what's available to us, and secondly, they're available in a portable form, which we know is very useful when it comes to testing things that are either awkward to move or are very fragile. Reflectance spectroscopy will be our main focus and is the less invasive of the two, but Raman spectroscopy has the benefit of being able to produce a spectra that acts as a pigment-specific fingerprint. Here are some examples of Raman spectra from a 2001 paper that identify 8 different pigments from an 800-year-old Bible in Paris, where the detected intensity of the spectra has been plotted as a function of wave number, which we know is a function of wavelength. So we can see here lapis lazuli has been identified, along with many others including azurite. Briefly, let's talk about Raman spectroscopy and how it's performed. Well, first, a sample is illuminated with monochromatic light from a laser. When photons scatter at the interface between the air and the pigment, most will scatter elastically, but a small fraction will scatter inelastically, which is what we're interested in. This is because of transitions in the vibrational modes of pigment molecules, which can either increase or decrease the energy and hence the wavelength of each photon involved in the inelastic scattering event. These transitions are pigment specific and are dictated by the potentials between its constituent atoms, producing spectra that acts as a pigment-specific fingerprint, making it very reliable for detecting the correct pigment. It's relatively non-invasive compared to non-optical methods, as we know, but on the downside, because the proportion of photons that are actually scattered inelastically is very low, actually as low as 0.01% of all incident photons, the number of photons that have to be incident on the sample when taking the reading has to be higher than it would when using other optical methods like reflectance spectroscopy. This means that the light intensity has to be high, or the exposure time has to be relatively long, or both. These both have the same desired effect of increasing the number of incident photons, so increasing the number of scattering events, but shining high-intensity laser light in a concentrated area for a prolonged period of time can lead to localized heating, which can be destructive for artifacts. So, this is what the plan is for the majority of our project in 3 main steps. The initial step is to create samples that we will then test by collecting the reflectance spectra from it, and then finally we will analyse the spectra and from which we will attempt to identify what pigments are present. So now going into what I mean by each of these 3 steps in more detail. For making the samples, we were all sent the same type of card, but 2 of us had one brand of paints and the other 2 had another. This here is the set I was sent. So we recently had a meeting to decide what size our samples should be, how we'll label them, how we should make them, and so far I've created one sample for each colour with 1 layer of paint, but we plan on experimenting with adding black to create shades, white to create tints, watering down the paint and seeing what effect that has on the spectra as well. We also plan to experiment with mixtures and layers. I think it will be interesting to see what we find with layering. My prediction is that if we layer different paints on top of each other before they dry, we will be able to detect a significant amount of the pigment beneath. I'm also interested to find out how much variation we find in spectra taken from paints that are supposed to be the same colour, but are different brands, or even if they're the same brand, but from a different batch. So the spectrometer that we have access to is the Ocean Optics Flame Miniature Spectrometer that detects reflectance spectra in the UV visible range. So let's have a look at how it works and how we can collect spectra. First, something called a reflectance standard is used to calibrate the spectrometer. Because the mechanism in which we were able to see the hue of pigments is subtractive colour, it makes sense to illuminate it with white light, so an appropriate reflectance standard to use would be white. Here is an example of what I mean. So we can see that for this specific reflectance standard, the reflectance spectra for the visible range sits around the same level, which is what we want. The data that we can then collect from our sample can be measured as a percentage of the intensity of the spectra from the reflectance standard for each wavelength. Looking at this graph, it's almost flat. In reality, it isn't an infinite number of infinitely thin peaks, but a sum of approximate Gaussians. So if you think of it, it's more the sum of the reflectance spectra of its constituents. So it would be more like this, but obviously it would be nowhere near as uniform. So this is why it's very difficult to get a flat spectrum, which is what you would be striving for if you wanted a perfect white reflectance standard. So now looking at the layout, we can see here that the light is scattered off a sample after being illuminated by an appropriate light source through a fibre optic cable. So first, the light travels down here, reaches the sample where the reflected light will be detected, and will travel back up here towards the spectrometer. So what happens once the light reaches the spectrometer? The light will then reach the lens from the fibre optic cable, where it is focused and then reflects off a collimating mirror, then towards a diffraction grating, which will split the light up into its constituent parts. Then it bounces off a curved mirror to maximise its resolution, where the CCD will then detect the light intensity. CCDs don't detect the energy of individual photons, only the intensity of light incident on it, so that's proportional to the number of photons. But with the nature of this setup, as the light is split up once it reaches the detector, different parts of the detector will correspond to different wavelengths, meaning that we can collect data in the form of intensity as a function of wavelength. So basically, automatically have a reflectance spectra. To show an example of what our results may look like and how we will go about identifying pigments, I'm going to introduce two studies that have both used reflectance spectroscopy to identify pigments and dyes. The first image shows the reflectance spectra collected separately from different green pigments. We can see how they all have a maximum in a similar position, and each line follows the same sort of trend. But as we stray from the visible region, the trends of these lines tend to diverge and don't hold many similarities. Now looking at a mixture, here it is. The solid line gives us the spectra taken straight from the mixture, and the other two lines show the reflectance spectra that add to make the resulting spectra of the mixture's constituents. As we can see, when there's a mixture, the resulting spectra becomes much more difficult to interpret, which is not the case with Raman spectroscopy. In this particular case, we have to look for the positions of absorption bands rather than the reflectance peaks, as you can see with the two vertical lines added to the graph. Now moving on to the second paper, this one is interesting because it helps to resolve the issue of mixtures identified in the last paper. So it talks about a program that successfully resolves pigments from a resultant spectra automatically, the majority of the time. It isn't as successful as the results obtained from an expert, but is far superior to just using a pigment library. It can also be operated by a non-expert. It has reasonable accuracy and is much quicker than the other two methods. This figure here, that I've taken from the paper, shows the three methods side by side used to identify pigments from a painting of a nativity scene from 1340. The automated program didn't detect as many as the expertly operated software, but was far superior to the pigment library from what pigments were available to reference. This links perfectly into what the primary aim of our research project is, and I will come back to that in a minute. Before I finish, I just want to share some final thoughts about what I found the most interesting so far. This project is all about colour, but even after learning about what causes colour, I'm left with more questions than I had before about what colour actually is. To begin with, I thought of colour as being caused simply by the reflectance spectra of pigments, but now, after comparing the spectra of pigments that are very close in colour, their reflectance spectra can differ dramatically, as long as the peak found in the visible range is in around the same place. Its spectra in any other part of the EM spectrum can be completely different, yet we still call it the same colour. Because of this, I've realised that even though the absolute hue of a pigment can be determined by looking at the spectrum in the 400 to 700 nanometer range, colour isn't actually a real physical thing, more of a concept that is a consequence of our biology and perception, and is much more subjective than we like to think. And so if colour is just a property that produces a sensation, then what things would be the same colour if the cones in our eyes were adapted to detect a completely different part of the light spectrum? I wonder what uranium would look like if our vision was tailored to see different hues of gamma. To bring things to a close, the final aim of this project will be to produce an extensive reference library of different pigments that can be used in the art history field to aid in restoration, preservation and authentication. But along the way, we will understand how both reflectance and Raman spectroscopy work and how to perform them. And finally, I hope to gain a detailed understanding of the physics of colour. Thank you for listening to my talk.